Hey everyone, my name is Michael Gage, and I'm a bioinformatics master's student at the University of Maine. In this presentation, we will be discussing an engineering advancement in 3D genomics known as Hi-C. Not that one. Let's begin with some background. The genome in the mammalian nucleus comprises several three-dimensional interactions that are pivotal in the functionality of the cell. These interactions are multitudinous, often comprising of near infinite loci-to-loci relationships. The introduction of Hi-C technology has allowed scientists to identify and map these relationships in the complete nuclear genome. As the first unbiased approach evaluating chromatin conformation, Hi-C has led researchers to unearth an array of gene regulation mechanisms. The Hi-C method was developed by Erez Lieberman-Aden and his colleagues at the Broad Institute in 2009. Lieberman studied mathematics, physics, and philosophy at Princeton, and later obtained a joint PhD in mathematics and bioengineering at the Harvard-MIT Division of Health Sciences and Technology. Currently, Lieberman works as a professor and researcher in molecular and human genetics at the Baylor School of Medicine. Prior to Hi-C, three key methods were employed for the small-scale analysis of genetic structure. These are 3C, 4C, and 5C. In the 3C technique, a single pair of chromosome loci can be contrasted. 4C utilizes inverse PCR to determine the interactions between one locus and all other loci, while 5C utilizes multiplex PCR to contrast all interactions within a collection of about 1,000 loci. These developmental stages in 3D genomics lacked a comprehensive approach to chromatin architecture and focused on pinpoint attributes of nucleic genome structure. Lieberman and his colleagues built upon these methods with the development of Hi-C, a new approach that does not specify a limited number of loci, but rather the entire nuclear genome. This refinement has led to a modern understanding of 3D nuclear genome architecture. The methodology of Hi-C combines both wet lab and computational methods to determine the architecture of nuclear chromatin. Cells are initially placed in a formaldehyde-fixing solution, cross-linking DNA to DNA interactions. DNA is then lysed and subsequently fragmented with the restriction endonuclease HIND3. After fragmentation, the 5' overhangs are filled in and labeled with a biotinylated nucleic acid. Blunt end ligation is performed at the site of the biotinylated nucleic acid, the ligation junction, to create a hybrid DNA strand. The cross-linking is then reversed and the samples are sheared. Streptavidin beads are bound to these linear fragments as they maintain a high affinity for biotin. This allows for the separation of biotinylated and unwanted DNA fragments. Once purified, the samples contain ligated DNA fragments that were physically adjacent in the original chromatin structure. These fragments are sequenced and computationally analyzed to visualize spatial adjacency in the genome. The output of such analysis can be visualized in heat maps and 3D architectural tools. Hi-C is advantageous in detecting all-to-all DNA interactions. However, it may not be effective in the precise study of individual locus-to-locus interactions like 3C or other molecular interactions. ChIA-PET, for example, analyzes specific protein-to-DNA interactions that may be desired by researchers. Today, Hi-C has been further refined to address some of these shortcomings. One refinement, known as Capture Hi-C, maintains similar methodology but allows for a more focused analysis of specific target regions of nuclear chromatin. Now that we've seen the methodology and shortcomings of Hi-C in relation to other technologies, let's look at some practical uses of this technology today. In a 2023 study on neuronal DNA and neurodegeneration, researchers utilized Hi-C to discover unresolved double-strand breaks that lead to genome structural variations. These variations altered the 3D genome and resulted in loss and gain of long-range interactions. Hi-C performed on neurons showed a decreased number of chromatin loops and a significant decrease in topologically associated domain, or TAD, integrity. TADs are self-interacting genomic regions, so the decrease in their integrity results in three-dimensional conformational changes in the chromatin structure. In another 2024 study, researchers utilized Hi-C to determine the presence of clinically relevant gene fusions in pediatric leukemia patients. They found a significant clinically relevant ratio of gene fusions in samples. They also determined that Hi-C provides a unique benefit in that it is useful in analyzing formalin-fixed, paraffin-embedded solid tissue that has been archived for 9 to 13 years. Typical RNA-seq methods perform poorly on such tissue due to the RNA degradation in tissue stored for prolonged periods of time. In conclusion to this presentation, I want to provide a brief summary of the important points we have visited during this exhibition of Hi-C. The genome in the nucleus comprises several three-dimensional interactions that are pivotal in the functionality of the cell. The introduction of Hi-C technology has allowed scientists to identify and map these relationships in the complete nuclear genome. Prior to Hi-C, three key methods were employed for the small-scale analysis of genetic structure. These are 3C, 4C, and 5C. These methods maintain a small-scale focus, but the need for full genome analysis still remained. The Hi-C method was developed by Aras Lieberman-Aden and his colleagues at the Broad Institute and allows researchers to analyze and visualize complete 3D genome architecture. The Hi-C methodology involves DNA to DNA cross-linking, lysing and fragmentation, biotin labeling, blunt end ligation, shearing and streptavidin bead binding, sequencing, and computational analysis. In comparison to methods like 3C or ChIA-PET, Hi-C does not function to analyze specific locus-to-locus interactions or DNA to protein interactions. However, the method is being actively refined to accomplish such tasks. And lastly, Hi-C has been utilized to determine genetic factors involved in pediatric leukemia, as well as the development of neurodegenerative diseases. Thank you.